Assalamu alaikum everyone. Bushra Umer from presenter teams in Biotechnology Association is here. And today we are going to discuss a really amazing topic, which is microfluidics. And we are also going to learn that how a single drop can brought the revolution. And we are going to link this technology, particularly with the life sciences. So let's see what's next. So here is the overview of what we are going to learn in this lecture. Some basics, working principles, examples, type of devices, and everything we can cover in this lecture. Okay, so before going to learn that what is microfluidics, let's see what is literature saying about this technology. So there's a quote by Mr. Frederick Bellegetti, who is one of the pioneers of microfluidics. And he's saying that rather than doing experiments in test tube and clinical flask, we can do bioscience on a chip. So why he thought like this? Because when we perform an experiment on a chip or on a small space, so we are reducing the time, since it is easy to control the sample which is present on a small amount. So we are reducing the time taking to perform that experiment. We are also reducing the size of the sample. Since chip is very small, so a small sample size is required, and we are also reducing the reagents that are being used in that particular experiment. So yeah, it is fascinating to use a chip for life sciences experiments. So this is the main thing, and we are going to learn that what is microfluidics. So first of all, when we divide the term into two, micro and fluidics. So basically the word micro means the size of 10 exponent minus 6, but here we are not limiting the size of fluidics, or fluidics means the fluid state of matter, and we are not limiting the size of fluids to micro size only. We are, as I mentioned here, that we can take femtoliters of sample, like femto is 10 exponent minus 15. Such a minute quantity can be taken to perform a certain kind of experiments. So this is the wonderful thing about use of microfluidics. And manipulation of small volumes, we will learn that what is the manipulation of fluids and through microchannels. So yeah, understand this thing with the help of this diagram. So we can see that a sample is introducing from this side, right? It's a very small drop. Then this will flow through these, you can say, very precise and concise channels. These are known as microchannels, and microchannels help the sample to run under proper conditions, temperature, pressure, and then it will reach to a place where the purpose of our experiment, like if we want to detect certain chemical, so microchannels will bring the sample to that area, and then will be a sensor or anything is present which will detect the compound we are interested in. So this is how microfluidics work on a chip. So here we are going to learn the working principle of microfluidics. Since I mentioned that microfluidics is used for different purposes, like we can use it for the identification of a certain type of compound, or we can use it to separate uh, compounds from a sample. So different compounds from a sample. So that's why I mentioned here it is purpose specific. So in this example, we are uh, seeing that a gas sample is used, and we are going to separate the volatile organic compounds from that. this is the specific purpose. So the basic three steps are these: miniaturization, integration, and sensing array. Miniaturization means that before introducing the sample, we have to prepare the microfluidic chip with miniature or small channels, like we learned earlier, microchannels. Then integration. The sample we are introducing in the microchannels must be integrated with its reagents properly so that we can get good results. And then the sensing array. Sensing array is, as I mentioned in the diagram previously, is a kind of result showing device. So there must be a sensing array or a sensing device which will process and proceed the integrated components to the computer. And then in this experiment, basically the VOC or volatile organic compounds are being identified, such as benzene, toluene. So this sensing array will help in identification of geometry of that organic compounds. And then with by identification, we are able to learn that which volatile organic compounds are present in that sample. So this is how biofluidic devices work in general, and specifically in this example of a gas sample. So this is the design of microfluidic design. We have already learned about the microchannels, inlet and outlet. So we see that microchannels are important, and the chip on which these microchannels are present is made up of PDMS or polydimethyl siloxane. This material is much like of plastic. And why we use PDMS specifically? Because this is transparent, and this enable us to watch our experiment directly. I mean, there is no such barrier in visualizing the experiment that is performing. So this is also a beauty of microfluidic, that we can directly observe what's going on in a particular experiment. And and yeah, there are certain additional properties, optical, electrical, and mechanical properties, that it can bear a specific amount of pressures, and also current and different other things that are required by experiment. And here is the product or microfluidic device. Inlet, outlet. Inlet is the point where the sample is introduced, and outlet where the sample or the experimental material leave the device. And these are the certain microchannels. So yeah, that's all about the design of microfluidic devices. So these are the few types of microfluidic devices. Number one is Y channel. As we can see that it is Y shaped, and we use this kind of microfluidic device when we need to separate two different components. And the second one is T junction, and we use this kind of microfluidic device when we need to prepare droplets of two different type of substances. So just like oil and water. So these are of different densities. So in T junction, we use this technique and we produce different type of drops, right? And the next one is EP or electric potential based, means simply this microfluidic device work on the principle of the flow of charges or electric current. And the last one is capillary action based microfluidic device. And in this type, we do not need any external pressure. We just simply introduce the sample through the channel, hydrophilic channel, and it flow by its own, just like we observe this thing in the capillary action. No need of external pressure. So here comes the applications of microfluidic technology and devices. Number one is lab on a chip. So lab on a chip means to perform experiment on a single microscopic chip, and this is the main thing in point of care diagnostics, which means when the experiment or some testing is done within the patient's unit, and we do not need a large lab or certain type of lab to perform these type of experiments. So here we are taking the example of blood, and this approach is used to identify a certain set of disease. And how we can do this? See, this is the inlet from where the sample is introduced, and then reagents, buffers are used, and this is multidimensional. Splitting microfluidic chip means there are, as you can see, the splitting is mentioned here. It's like here the sample is introduced from this, and then it goes here mixed with the lysis buffer, and then it travel our after mixing, and then PCR reagent is uh, added to it, then again mixing or amplification occur, and then PCR. Why PCR or polymerase chain reaction is the amplification or making copies of particular set of genes. So all this process occur by different buffers and reagents and dyes, and then after adding dyes, basically this dye or prop dye is the outlet procedure, and then after this we are going to observe the SNPs, which is single nucleotide polymorphism, and these are the set of genes which shows the, you can say, variations or genetic variations. So SNP help in the diagnosis of different type of diseases. So this is how lab on a chip work, and from here the rest of the sample is excluded from the chip, and what we need is SNP detection. Our purpose of experiment is completed here, and rest of the things are moved out. So this is how lab on a chip works. The next is, so this is another interesting application of microfluidics, which is organ on a chip. So particularly we took muscle example in this regard, and why to use it? As I mentioned, diabetes, obesity, and different type of cancer can cause wasting of the muscle. Wasting means that there is a degradation in strength of a muscle due to a disease. So to identify that how the muscle is deteriorating and how its function is destroyed, we use organ in a chip technology. And what we do in this, we take a small amount of muscle, we process it, and then an isotropic non-uniform design means we take different readings or different measurements or lengths of the muscle, and then we test that how that muscle become weak, and we can also learn that what type of drug we can use to activate that muscle, and how that drug is acting, and all these things are occurring on a single chip. So this is the wonder of microfluidic technology, and basically microengineering biomimicry of the human body emulates means while using organ on a chip technology, we are mimicking or copying the function, the actual function of muscle, which is to carry the nerve impulse and all the things outside the body. on a chip. So this is all about organ in a chip. There are some other applications of microfluidic technology. We have learned about the example of volatile organic compounds detection, uh, which come under these things, or environmental contaminants detection. And we can also use the pressure and other means like electric potential, voltage, magnetic field for the separation of charges, like bacterial electrode transfer, gene transfer, and different charge generations, to study the different behaviors of bacteria. And biofilm formation is one of that fascinating application. Biofilm formation occur when bacteria favor some, sorry, face some unfavorable conditions. So it is an alternative life form. So by using microfluidic technology, we can also learn and study how certain bacteria and what under what pressure bacteria are forming the biofilms. Okay, so we have learned the examples of microfluidic technology, but there are certain pros and cons of every technology. So let's see. As we learned that there are numerous advantages of this technology: less time required and less amount of sample is required. It can be done on a small space, and reagents, chemicals, and the cost of these things are reduced. And the cons or the disadvantages are that sometimes it is not possible to maintain the pressure throughout the experiment. So this is a disadvantage. And difficult to standardize means there is no standard quantity required or known to carry out experiment for the first time. And the stress, shear stress, and the pressure imbalance can cause the degradation of the cells or the sample that is introduced in the microfluidic chip. So this can be a disadvantage. So here comes the end. This is the summary, and we can say that there is a lot of potential in the field of microfluidics, and by developing more knowledge about it, like more sensing techniques and other things, and applying artificial intelligence in this thing too, we can use the technology with more advancements and to carry out our experiments on a greater extent by using this technology. Yes, this can also help in the formation of personalized medicine, because we can identify the certain genomic structure or genomic content of the organism of the human being. So we can describe or give the proper medicine by using microfluidics, and the possibilities are endless. Yes, we can say. So these are the references from where I prepared this lecture. Thank you so much for listening. I'm waiting for your queries. Mention them in the comments. Thank you so much.